Assalamu alaikum, everyone. My name is Khalid Jamiel. I'm a fourth-year medical student, and today I'm going to be giving the immunology crash course. This is going to cover lectures 1 to 8, which are the lectures included in your midterm. If you have any questions or you find any mistakes or anything, please don't hesitate to text me or call me anytime. Inshallah, it's going to be an easy crash course. Immunology is a very easy topic. However, what makes it difficult is that the slides are very condensed and compact. And so if we break down the topics, it's actually going to be very easy. And inshallah, by the end of the crash course, you're also going to realize that it is, in fact, very easy. Now, the questions in the exam are usually simple. However, they usually test your understanding rather than your memorization. So you need to understand everything you read very well. There's no point of skipping a point if you don't understand it, because Dr. Darwin is very clever at testing your understanding. However, it's very easy, inshallah. So let's get started. I always indicate when I start a new lecture on the top left corner. So that way you can follow up with your slides if you want. Okay. So let's go back to the very basics. All blood cells come from a common cell called the hematopoietic stem cell. Hematopoietic stem cell gives rise to a myeloid progenitor and a lymphoid progenitor. The myeloid progenitor gives all of those cells, monocytes, basophils, neutrophils, eosinophils, and a couple of other cells that are not relevant to the immunology course, like red blood cells and thrombocytes and so on. The lymphoid progenitor gives rise to natural killer cells as well as T and B cells. Now, everything that's in orange has to do with the innate immune system. Everything over here, which are basically T and B lymphocytes, have to do with the adaptive immune system. And I'm guessing you guys know all of this already. So I'm gonna go quickly through the differences between the innate and the adaptive immunity. So the innate immunity, by definition, is present since birth. And the response time for the innate immunity is immediate. It's not specific to any antigen or any microbe. And the same response happens to each subsequent infection. So, for example, if I was infected with a certain strain of Staph aureus today, and then 1 month later again, the innate response to that infection is going to be exactly the same. As for the adaptive immunity, it requires prior exposure to antigen. So if I was infected for the first time with the Staph aureus strain today, there wouldn't be any adaptive immune cells ready to attack that Staph aureus. So it needs prior exposure, and because it needs to adapt, it's slower, and it's antigen specific, obviously. And then there's stronger response to subsequent infections because of the presence of memory cells. I'm going fast over here because this is already stuff you should know. Now, let's talk more in detail about the innate immune system. So the innate immune system isn't simply just the cells we mentioned. It has many components. The anatomical component of the innate immune system includes the skin, tight junctions between the epithelium, mucous membranes that contain cilia and mucus to trap any incoming infections, and then flushing processes like tears, saliva, all of those are used to flush out infections. The biochemical component includes fatty acids, or sebum, and sweat, and these are contain antimicrobials that can kill infections. The acidic pH of the stomach, vagina, and other areas of the body are also or can also contribute to microbial killing. Then we have lysozymes and phospholipases, which are antimicrobials that are found in the saliva and tears. And finally, we have the lung surfactant, which also contributes to the immunity of the lung. And then for the biological components, we have the normal microflora or the normal microbiota, and these help in the innate immune system by, number one, competing for nutrients with the infectious bacteria. Secondly, they also take up space, so they don't allow the bacteria to colonize. And number three, they also can produce antimicrobials. Now, examples of these antimicrobials are phospholipases, defensins, and ribonucleases. Now, a general rule to follow in my slides, if something is in lowercase, that means it's probably not important and it's probably not going to be asked about. It's just there for the sake of completion. This whole slide is there for the sake of completion, honestly. And as for blood components, we have acute phase reactants. These are proteins produced from the liver and are upregulated when there's inflammation, and they themselves upregulate inflammation in order to get rid of the bacteria faster. The most important one for this course is mannose binding lectin, and you'll know why later on. Complements and cytokines also contribute. Lactoferrin, ceruloplasmin also contribute by taking up iron. Bacteria needs iron to grow, and so they prevent bacteria from growing. And then the coagulation system also helps. You don't need to go into any details. It's just a general slide. That's it for the first lecture. Now, to talk more about the innate immune system, how do the innate immune system cells recognize when they need to start the inflammation or start working? So the first and most important thing is this reaction over here, a reaction between PAMPs and PRRs. PAMP stands for PAMP stands for pattern associated molecular patterns, and PRR stands for pattern recognition receptors. PAMPs are present in pathogens, and PRRs are receptors found in innate immune cells. Okay. Now, PAMPs can come in many forms. The most common form is called MAPS, MAMPS, and this stands for microbial associated molecular pattern. And by the name, you know that these MAMPs are present in bacteria or microbes in general. The most common MAMP is LPS. Now, because MAMPs are the most common type of PAMPs, when we say PAMPs, we usually are referring to MAMPs. However, PAMPs do have other subclassifications under them, and we're going to talk about that soon. Now, what happens when a PAMP is recognized by a PRR or a pattern recognition receptor? There are also many types of pattern recognition receptors, and we're going to talk more about those soon. But the main three things that happen when this interaction happens is number one, phagocytosis. So number one, phagocytosis. Number two, there's production of cytokines, inflammatory cytokines, in order to enhance inflammation, cytokines. And this is what starts acute inflammation. You should know acute inflammation well from your pathology lectures. Number three is lymphocyte activation. And this is the activation of the adaptive immunity. So the innate immunity does have to do with adaptive immunity. Actually, one of the main functions of the innate immunity is to activate adaptive immunity. Okay, so let's read what I said in text. PAMPs, or pattern associated molecular patterns, there are two types. There are MAMPs, and those are the most common type. And the most common example is LPS, found in gram-negative, and teichoic acid, found in gram-positive bacteria. Then we have other types of PAMPs called DAMPs. And these are important for something we call sterile inflammation. And we're going to talk about this soon. Okay. Then we have the main example of DAMPs are alarmins. However, any part of the cell that when the cell dies, any part of the cell that leaves the cell is recognized as a DAMP. So it's DNA, RNA, intracellular proteins, anything that's not supposed to be outside the cell is recognized as a DAMP. Okay. Now for the PRRs, we have three main types. The main type, and the one that you're probably going to be asked about, is the TLR, and this responds to MAPs. Okay, so TLRs go with MAPs, and they're extracellular. Okay, now PRRs can be extracellular or intracellular. That's why you need to know which ones are extra and which ones are intracellular. Node-like receptors are intracellular, and they recognize DAPs. Okay, and fungal complex sugars, but this is not important. So what happens? So this is a cell, and then the node-like receptors are actually inside the cell. When the cell, when something that's not supposed to be in the cytoplasm enters the cell, and binds with this node-like receptor. This node-like receptor is going to combine with other node-like receptors and form something called an inflammasome. This complex is called inflammasome. These inflammasomes are going to act as cleavage protein, and they're going to go and break down zymogens of IL-1 and IL-17, activating them. Zymogens are basically proproteins. A proprotein is a protein that needs cleavage in order to be activated. Okay, so NLR or node-like receptors are important for sterile inflammation. Sterile inflammation means inflammation without bacteria being present. Obviously, when cells die, there is inflammation always. When there's trauma, there is inflammation. There doesn't have to be necessarily a bacteria or something in order to activate inflammation. It can be sterile, and this is how it's done. IL-1 and IL-18 then go ahead and activate acute inflammation. Then we have Rig-like receptors, and probably the significance of this is that it's intracellular and therefore recognizes viral DNA. And then obviously activates inflammation as well. But you don't need to know details. However, you need to know this, the inflammasome formation and IL-1, IL-18 activation. And this, we said, is the main thing we are focusing on in this course, which is when MAMP activates TLR. Okay, phagocytosis of the bacteria, release of cytokines, and then antigen presenting to activate adaptive immunity. This slide is very important. Okay, so we spoke about the anatomical, the biochemical, the biological components of the innate immunity. However, the most important component, obviously, is the cellular component. And we can classify the cellular component of innate immunity by functions. So the first function is phagocytosis. And we have three main phagocytes. Those are neutrophils, macrophages or monocytes, and dendritic cells. So an important fact about neutrophils is that when they phagocytose a bacteria, they obviously throw the bacteria away, but then some of the neutrophils die. And then when the neutrophils die, طبعا neutrophils do not activate the adaptive immune system. This is a separate function called antigen presenting. So neutrophils phagocytose, although they phagocytose, they cannot activate the adaptive immune system. This is something to keep in mind. Now, neutrophils, when they die, they release nets. And nets are molecules that simply alert the surrounding cells that there's a damaging process going on, and that activates a couple of things that enhance immunity. That's all you need to know about nets. Now, macrophages or monocytes, the main thing you need to know is that on the surface of macrophages, they have CD14. CD14 is associated with TLRs. They're bound together. Okay, so there's an association between CD14 of macrophages or monocytes and TLRs. And then we have dendritic cells, which are basically cells that have a lot of projections like this. They're professional phagocytotic and antigen presenting cells. They're very important in the activation of adaptive immune system. That's their main function. Activation of adaptive immune system. Now, obviously, we know that when monocytes are in the blood, they're called monocytes, but then when they live in the tissue, they're called macrophages. Macrophages have many names, like, for example, when they live in the liver, they're called Kupffer cells. When they live in the brain, they're called macroglia. When they live in the skin, they're called Langerhans cells. When they live in the lung, they're called alveolar macrophages, and so on. So it depends on where they live. However, monocytes and macrophages are basically the same cell. Now, for antigen presenting, and this is a very important function, and the main implementation of this is to activate the adaptive immune system. And the main cells are macrophages, dendritic cells in the innate immune system. However, B cells can also be antigen presenting cells. Okay, and we'll talk more about this in detail later. However, B cells are not part of the innate immunity. They're not part of the innate immunity, but can also function in antigen presenting cells. Now, the main point of confusion here comes, people, first of all, think, because neutrophils are phagocytotic cells, they can antigen they can present antigens. That's wrong. And people also think that only innate immune cells can antigen present. However, B cells are also antigen presenting cells. Keep that in mind. Now, another function is cytotoxicity, which is simply directly killing the cells. And the only cell in the innate immune system that can do this are NK cells. Their marker is CD56, and their main function is to kill viruses and cancer cells. Now, this is a general rule to follow. Whenever a cell kills directly, its main function is going to be viruses and cancer cells. That's because viruses infect the cells. They're not roaming outside like bacteria. And so you need to kill the cells that have the viruses in order to prevent the spreading of this virus. Cancer cells always have weird proteins and weird surface proteins. That's how NK cells can know that this is a cancer cell and then kills the cancer cell, trying to prevent the tumor from happening. Okay? Finally, we have allergy or hypersensitivity, and this is mediated by eosinophils, mast cells, and basophils. There isn't much detail about this that you need to know for the midterm. I hope this slide is clear. طيب. Now, the second lecture is done. We're jumping into adaptive immunities. So, to give an overview of the adaptive immune system, we need to know the basics first. We have two types of lymphoid structures. Now, from the word lymphoid, wherever there's lymphoid or lymphocyte or whatever, it has to do with the adaptive immune system, okay? So we have two lymphoid structures, two types, primary lymphoid structures and secondary lymphoid structures. Primary lymphoid structures, by definition, are the site of production and actually, this should be maturation. So please fix this in your slides. This should be maturation. Activation is wrong, okay? So primary lymphoid structures are the sites of either production or maturation of lymphocytes. The bone marrow is the site of production of all lymphocytes, so both B and T lymphocytes. However, it's the site of maturation of only B cells. So where do T cells mature? They mature in the thymus. This is actually where the name of the cell comes from. B is for bone marrow and T is for thymus. It's for the place where they mature. This is not a mnemonic. It's actually why they call it B or T cells. Now, for the secondary lymphoid structures, this is the site of activation of lymphocytes. And the main secondary lymphoid structure is lymph nodes. The main one is lymph nodes. Other examples are the spleen and the mucosa-associated lymphoid tissue. This is not important at all. Spleen is kind of important, but lymph nodes is the most important. And we're going to talk about how these lymphocytes get activated later on. However, just know for now that primary lymphoid structure equals production and maturation. Secondary lymphoid structure equals activation. So we have two components of the adaptive immunity, or two types of adaptive immunity, let's say. We have the humoral immunity and cell-mediated immunity. Cell-mediated immunity means direct cell killing, and this is done by cytotoxic T cells. Humoral immunity is antibody-mediated, and this is done by B cells. And there's another type of T cells called helper T cells, and these are more involved in activating this. Okay, so let's look at the diagram. What happens is an antigen presenting cell, and remember there are three types, the macrophages, the dendritic cells, or the B cells, carry the antigen in a receptor called MHC, and we're going to talk much more about this later on, so don't panic. This is going to react with helper T cells or CD4 positive T cells, and this is gonna activate B cells in order to produce antibodies specific to this pathogen that has this antigen. And antibodies can kill bacteria in many ways that we are gonna discuss in a bit. The second type of adaptive immunity is just direct cell killing. T cells, they go around the CD8 positive or the cytotoxic T cells. They go around sniffing human normal cells. And if they sense that something is wrong, they're gonna send perforin and granzyme, which are lytic enzymes that break down the cell membrane of the cell, and that will lead to the lysis or reversing of the cell. So this is the basic overview of adaptive immunity. Now, in each single line, we're gonna go into so much detail that you're gonna hate immunology for sure. So this is an oversimplification. Okay, don't think it's so. First of all, let's talk about antibodies in detail. Okay, now the antibody structure. This slide is heavy, but it's extremely important and guaranteed. There's going to be questions about this in the midterm and the final. So you need to know the structure of the antibody very, very well. Okay, so let's go down to the basics. Each antibody is made of 4 chains. Okay, 2 identical heavy chains and 2 identical light chains. Identical. Okay, so how do they coexist like this? We have these 2 heavy chains. So this is one, this is the second one. They're identical. Right? And these are the light chains. And this is how they exist in nature. Okay. Now, each chain, whether heavy or light, has a constant region and a variable region. Okay, or a constant domain and a variable domain. Okay, so all of this in dark blue till here is the constant domain of the heavy chain. Okay, and this dark red is the constant domain of the light chain, Cl or Ch. And then for the variable domains, it's this light red and light blue. Okay. I hope I didn't lose you so far. So simple. We have 2 identical heavy chains and 2 identical light chains. And each chain has a constant domain and a variable domain. Okay. Now let's talk about regions. Regions are different from domains. They're not the same. Okay. Do not equal domain. Regions is this whole thing. This box is called the region, and this box is called the region. Well, this is a domain, this is a domain, this is the domain, this is the domain. Okay. So when I say constant region or FC, I mean this whole box. When I say constant domain, I mean just this or just this. Okay. This is the constant region, or also called FC portion of the antibody. This is the it's called Fab. I don't know what Fab means, but it's called the Fab region. Okay. Now FC portion, or the constant, and it's mostly formed by the constant domain of the heavy chain. Okay, this part over here is FC. It does not include the constant domain of the light chain. It's only the constant domain of the heavy chain. Okay, this part is called FC, and this determines the isotype of the antibody. So whether it's going to be IgM, IgG, IgE, depends on the FC. Okay, this is very important. Everything I'm saying right now is very important. So please make sure you understand. The Fab portion determines the specificity of the antibody to an antigen. Okay, so this region is what determines what the antibody is going to be specific to. Okay, you can think of the antibody as a lock, and then the antigen as the key. Okay, so they need to fit perfectly in order for it to work, and this is called specificity. Okay, so each antibody is specific to one and only one antigen. And what determines which region is responsible for this specificity? It's the Fab region. And what's the FC region or FC portion responsible for? The isotype, or the type of the antibody. Okay, it's very important. Now, antibodies can exist in 2 forms. Okay, and okay, let's go back a bit. We have an N terminal in every single protein in the body. There's an N terminal and a C terminal. Okay, so this side is the N terminal of these proteins, and this side is the C terminal of these proteins. Okay, now antibodies exist in 2 types. Okay, they're either bound to B cells like this, and in this case they're called BCR, or B cell receptor. Okay, or they can be just roaming around in the blood. Okay, and those are just secreted antibodies. Okay, now what determines whether the antibody is going to be a BCR, or it's going to be secreted. The C terminal of the heavy chain of the constant region of the heavy chain. Okay, so the C terminal of the heavy chain determines whether it's going to be bound to the BCR to the B cell, or whether it's going to be secreted. Okay, this is also very important. Now, finally, up here in the N terminal, okay, in here, if we break down this portion, this end portion, okay, we have sequences of amino acids called CDRs. Okay, CDRs. CDR stands for complementary determining region. Okay, now these, we have 3 in each. We have 3 CDRs over here, and 3 CDRs over here, 3 CDRs over here, and 3 CDRs over here. Okay, now these have very specific sequence that determine the shape of the lock. As I said, you can think of the antibody as a lock. Okay, so it can either be this shape, this shape, this shape. What determines what shape the antibody lock is going to be? The CDR regions, okay, which are regions at the tip, at the end of the variable domain of both heavy and light chains. Now, as I said, we have 3 in each, صح? Now, CDR3, or the third region, is the most important one for the variability of antibodies and for the specificity of the antibody. Okay, again, we have 3 CDRs on each side. Okay, CDR3, the third one, is the most important one for the specificity of the antibody. Okay, that's all you need to know. If you can't imagine it, just memorize it. Okay, I hope the slide is clear. It's extremely important. Okay, one point I forgot: light chains, those light chains, this whole thing can exist in 2 forms as well. It can be kappa or lambda. You don't need to know what's the difference in kappa or lambda. You don't need to know anything. You just need to know that they exist in 2 forms. And this is important later on, okay? Now, there are rules that have to be applied to all antibodies. The chains, as I said, have to be identical. And you can't have a light chain here that's different from the light chain over here. You can't have مثلا هنا kappa light chain and هنا lambda light chain. Okay, you can't. You can't have a heavy chain that's for IgG and then the other heavy chain is for IgM. That doesn't work this way. Okay, they have to be exactly complementary in order for the structure to form. I hope the slide is clear. Very, very extremely important slide. Okay. Like, now there are concepts we have to understand. Affinity and avidity. Now, what happens is when an antibody binds to an antigen, the antigen is usually way bigger than the antigen binding site, as we can see here. So the specific part of the antigen that binds to the antigen binding site is called the epitope. Okay, this is the definition of the epitope. It's the part of the antigen that binds to the antigen specific part or the binding site in the antibody. Okay. Now, affinity. What does affinity mean? It means the strength of the binding between a single combining site of an antibody and the epitope of the antigen. Okay. So how strong the interaction is over here and how strong the binding is over here is called affinity. Okay. Now there's another important term, which is avidity. Avidity is the total number of epitopes an antibody binds at any time. Okay. So this is determined by the valency of the antibody isotype. What does this mean? How many antigens is this antibody binding? 2. So we call it bivalent. Okay. Some antibodies, however, exist in this form. Okay, we call them pentamers. Why? Because as you can see, there's 1, 2, 3, 4, 5 antibodies all joined together, and this is how it's secreted in the body. This is how it exists in the body. Okay, and they're all joined together by J protein, or the joining protein. Now, how many antibodies is this, and how many antigens is this antibody binding? Of 10. Okay, so the avidity here is much higher than the avidity here, okay? That's because this exists in pentamers. And this is what we call valency. Valency is how many antigens the antibody can bind in its natural existing form in the body. So antigens that exist like this, so they're bivalent. Antibodies that exist like this are bivalent. Antibodies that exist like this are pentamers, and therefore have much higher avidity than these. Okay, and you need to know which ones exist how. Okay, so IgG and IgE exist in the bivalent form. IgM and IgA. Oh, this is not bivalent. This is multivalent. Okay, let me fix this as well. Multivalent. Okay. IgM and IgA are multivalent antibodies. Okay. IgM exists in pentamers or hexamers sometimes. IgA exists in dimers. Okay, so two connected together, and so their valency is 4 antigens. I hope this point is clear. Okay, now we have different types of antigens. We have immunogens and haptens. Okay, so what do those mean? Immunogens are antigens that can stimulate an immune response. And this is because they're large enough to stimulate an immune response. Haptens are also antigens, but they cannot stimulate an immune response because they're not large enough. Okay, as I said, usually antigens are much larger than antigen binding site. If that's not the case, they're called haptens and and they're not immunogenic. They are we can't call them immunogens. Okay, and however, some haptens can be conjugated with a carrier protein, like in the lab. Sometimes when they want something to bind, they put with it a carrier protein. Okay, and the carrier protein increases its weight and makes it larger, therefore it becomes immunogenic. Okay, so the whole issue is with the size. If the size is big enough, it's an immunogen. If it's not big enough, it's a hapten. As simple as that. So I hope the slide is clear. It's a straightforward slide. Okay, now isotypes of antibodies. This is an extremely important slide, and it's easy marks. And there's a guaranteed question on this, and it's easy marks. You can get marks easy. So firstly, IgM, okay? This is the major antibody in the primary response, okay? So the first time the body is exposed to the bacteria or whatever, if you take the serum and see what antibody is raised, you'll find mostly IgM, okay? As you can see here, okay? The primary infection, the most thing that comes up in the beginning is IgM. Okay, and as I said, it exists in pentamers or hexamers. This is very important. It has low affinity, however, very high avidity. Okay, as we already explained. Now, IgG is the major antibody in secondary response. Okay, as we can see over here, when there's a secondary infection, IgG rises very fast and it peaks. Now, I guarantee you 100% there's going to be a question about this concept. 100%. They can ask, wallah, someone got an infection, and when they measured the serum, most of the immunoglobulins were IgG. Or what does that indicate? It indicates that this is a secondary infection. However, if it was mostly IgM, it indicates that this is a primary infection. Okay, so you need to understand this concept very well. They can even get this graph and cover IgG or cover IgM and ask you to label it. المهم, they can ask you in any way. However, this concept is going to be asked about for sure. Now, IgG has high affinity but low avidity. So it's basically the opposite of IgM. As I said, because it is bivalent. It exists بس كده as one. It's not a dimer or it's not multivalent. Okay. It has the longest half-life in the blood. This is also a very extremely important point. Okay, because why does it have a high half-life? That's because it can bind to something called the neonatal Fc receptor. Now, antibodies, they're broken down in cells. Okay, macrophages or whatever. they swallow the antibodies in order to break them down when they stay for too long. Okay. However, IgG can bind with a specific receptor on those cells that are going to break them down, called neonatal Fc receptor. This allows the IgG to simply go through the cell. So let's say this is the IgG. It can get endocytosed and exocytosed through the other side because it binds to this specific receptor called the neonatal Fc receptor. Okay. And okay, it obviously binds to the Fc portion. Okay, not side. So it's supposed to be flipped. It binds to the Fc portion, and then it can induce transcytosis, and therefore it's not degraded inside the cell. And that's why IgG has the highest or the longest half-life in the blood. Okay. Now, it can also move through the placenta. Again, this is due to the neonatal Fc receptor. This is very important. These two features are special for IgG. Okay, so it's important to remember whenever there's something unique. It's important to remember. Now, IgA is the most important in mucosal immunity. This can be asked in multiple ways. If I take a sample from your nose or from your mouth or from your throat, what would be the most abundant immunoglobulin? That would be IgA. Okay, and it's the most abundantly produced. However, it's not the most in the blood. You need to make a difference. What's the highest in the blood? It's usually IgG. Okay, because it has the longest half-life. What's the most produced in general? It's IgA. However, we don't find it in the blood because it's usually shed in the fluids, whether it was cough, sweat, through the GI tract. It's usually excreted. That's why we don't find it high in the blood, although it's the most one that's produced. IgE is important in allergic reactions, and you'll know later on as well, it's important in parasitic infections. And it has the shortest half-life in the blood. However, there's another form that's usually bound to mast cells or eosinophils, and this lasts for long. It does not have a short life. However, when it's just roaming around in the blood, it has the shortest half-life. And I like to remember this because I don't want the allergic reaction to go for too long, or else it can be harmful for the body. And that's why we need to get rid of the IgE quickly. Then IgD is the least important one. However, it has a very important significance that we're going to talk about again later. It's found as a BCR in naive B cells. So we don't find it roaming around in the blood. Never. Okay. If we find IgD, we find it in as a B cell receptor. And as if you can recall, what tells us if the antibody is going to be a BCR or secreted in the blood, it's the C-terminal of the heavy chain. Yeah, it's just a quick revision. Like, what are naive B cells? These are B cells that have not seen an infection before. And we're going to talk much more about this later on in detail, inshallah. Okay, so we're going to revisit this point. Don't worry if you don't understand it. I hope this slide is clear. It's a very important slide. Now, something I forgot to mention. Antibodies have many names. Okay, so if I say immunoglobulin, Ig, if I say gamma globulin, those are all names or different names for antibodies. Okay, so don't get confused in the exam. Like, what I said earlier that antibodies can kill pathogens in many ways. What are those ways? The first way is called opsonization. Okay, and this is a specific function of only IgG. Okay, so what does this do? It tags the pathogen for lysis. Okay, so this pathogen, for example, had this bound to it, and then the phagocytic cells like neutrophils, dendritic cells, and macrophages can recognize the Fc portion of this antibody and know this antibody is bound to a bacteria, and this induces the process of phagocytosis. So you can basically say it makes pathogens more tasty for phagocytic cells. Okay, it makes phagocytic cells be able to smell them from far away. So this is one way. The second way is neutralization, and this is mainly applicable for viruses. So viruses need to infect host cells in order to be functioning. However, antibodies can surround those viruses, not allowing them to bind with the cell with the host cell, and therefore not allowing the virus to become active. The third way is through the complement system, and we're going to talk much about this later on. The fourth way is through direct killing by natural killer cells. Okay, so natural killer cells have CD16 on their. I think I put CD56, folks. I think natural killer cells marker is CD16 and not CD56. If I put it as CD56 in the earlier slide, please fix it. So it has CD16, which is basically an FC receptor that tags the bacteria for direct killing through the secretion of perforin and granzyme, which lyses the bacteria. Okay. Now there's أنا صراحة I don't think this is important, but I just put it for the sake of completion. Polyclonal versus monoclonal antibodies. So how do they get antibodies for treatment? You know that antibodies are sometimes used for treatment. So polyclonal antibodies, they simply inject the bacteria with the antigen, and then they wait for the B cells to produce antibodies, and then they take these antibodies from the serum of the rabbit and they use it as a treatment. However, the more important application of antibodies is monoclonal antibodies. I'm sure you heard about this many times before. What they do is they take cells from the mouse, and then they take cancer cells, myeloma cells, which are hyperactive B cells, okay? And they bind them together, and they wait for the B cells to become specific to an antigen. And then they select only the B cells they want that are specific to the antigen they want. And they harvest these cells, and then they take the antibodies formed by these cells, and those are all specific for one antigen. Okay, they're all the same antibody, so they're specific for one antigen. And this is used in the identification of unique markers for diagnosis of cancer. It's also used as a therapy to target one protein or something, and that's how it can be used as a therapy. That's it for this lecture. I'll move on to lecture 4. This is a very tricky lecture, but inshallah, I'm trying to make it as simple as possible. Okay. So this lecture talks about how B cells develop and mature from the bone marrow. Okay. Before we go into how B cells develop and mature, a very important concept we need to understand is VDJ, recombination and junctional diversity. What is the goal of this? The goal of this is to produce a specific and functioning antibody. Okay. So if you ever think, طيب أنا عندي all antibodies have to be specific. How do we achieve this specificity? طيب حتى although antibodies have to be specific, we have millions and millions of pathogens. How am I supposed to produce this much variability in the antigen binding site? صح? So this question is answered while the B cell is developing in the bone marrow. The B cell is still immature, and that's when variability of the antigen binding site starts happening. Okay, so how does this happen? This is the DNA. Okay, this is a strand of DNA. Okay, either the paternal or the maternal strand. Okay, now each DNA of B cells specifically has segments called B segments, and then segments called D segments, and then segments called J segments. V stands for variable, D stands for diverse, and J stands for junctional. You don't need to know what they stand for. طيب. But know V, D, J segments, okay? So what happens is, at the transcriptional level, or at the DNA level of the B cell, first thing that happens is that there's recombination between the between the D and the J segments. Okay, random recombination. So it takes مثلا, let's say this is D1, this is D2, this is D3, this is D4, and then the same for J1, J2, J3. So the first thing that happens is an enzyme called RAG1, 2, RAG1 and RAG2. They cause random combination of D and J. So this may be D3 and D1 plus J1 and J2, or J1 and J3, okay? We have many J segments, many D segments. This is just a simplification, okay? The second step is, again, RAG1, RAG2 causes random recombination, but this time with the V as well. So the V combines with this DJ segments. So this might be V30, V22, V4, for example. And then these are the same as earlier, okay? So this is still at the DNA level. Now you'll realize over here there's a constant exome, okay? And I'll tell you why this is important later on, but this doesn't change, okay? It's constant in the B cell. It's the same in all B cells, actually, this constant exome, okay? Now, what happens is there's this enzyme called TDT. After this combination, random combination happens, TDT comes and then it adds or deletes nucleotides randomly from the ends of this sequence, okay? Just to make it even more random. So all of this is just to make it as variable as possible and as random as possible. Now this sequence then gets transcribed and spliced, okay, into, this is mRNA. So we're in DNA level, now we are in mRNA level, okay? Now you'll notice that there's only one V, only one D, and only one J, okay? And على أي أساس, or how is it chosen, and which V becomes this, which D becomes this, which J becomes this? It's also random, okay? So everything is random regarding the VDJ. And this sequence, mRNA sequence, then gets translated by ribosomes into a heavy chain, okay? This is one heavy chain, and this is the second heavy chain. As I said, the heavy chain has a constant region and then the variable domain. Constant domain, variable domain. طيب, where does the constant domain come from? It comes from this constant exome that's always been there and unchanged, okay? And it also came here. تمام? It's the same for every single B cell, this constant exome, okay? And what is this heavy chain? It's called the mu heavy chain. This mu heavy chain makes the antibody IgM, okay? So all developing B cells, the first antibody is IgM. And that's why it's always the antibody of the primary response, okay? As we said, this FC portion, since it's the same in all B cells, it's mu. That's why all B cells in the beginning have IgM. Right? This point is very important. However, this variable domain is extremely variable. Right? Let's count how many times it got randomized. Here, it got randomized. Then here, it got randomized even more. Then TDT came and messed up the whole thing, and it became randomized again. And then when transcribing and splicing, there's also another randomization until we reached here. And that's how we get the wide antibody repertoire, the word is, which means is we have a storage of antibodies that is very diverse, and it can bind to many antigens because of this VDJ recombination. So let's read what I said in text. The first step is DJ segment joining at the DNA level. The second step is V plus DJ segment joining again in DNA level, okay? And both of those processes are done by RAG1, 2 enzyme. Then we have a process called junctional diversity, which means TDT is gonna add sequences or nucleotides randomly to the end of the coding sequence. So here and here, it's gonna add or delete randomly to make it even more variable. Then transcription and splicing occurs. Now note that the constant exome is always mu. It's not gonna change, okay? And that's why all developing B cells start with IgM. Then this gets translated into the heavy chain, okay? Now, what about the light chain? That's a very valid question. The exact same process happens, however, there is no D segment. It's just V and J, okay? There's no D segment. However, the same exact process happens, okay, for light chains too. Right. Now we have two rules for DNA expression. The first thing is allelic exclusion, which simply means in a single B cell, either the maternal or the paternal genes are expressed, but not both, okay? So this DNA is from either the father or the mother. In the same B cell, uh, the mother DNA is never going to be expressed. It's only the father DNA. So this B cell has the exact same expression of the exact same DNA, and that's why all the antibodies of a single B cell is identical always. Okay. Now, as I mentioned earlier, each individual B cell can produce only one type of antibody containing only one isotype of heavy chain, only one, right? And only one type of light chain, either kappa or lambda. And these you don't need to know. All you need to know is mu is IgM. The rest you don't need to know what they stand for or what they will become. However, and this can be IgG, this can be IgA, you get the idea. So the type of the heavy chain determines what the isotype of the antibody is going to be. Right? So I hope you got this concept. It's very important. The enzyme names are very important. What they do exactly are very important. You also need to know what level everything is happening in. But this is the DNA level, this is the mRNA level, and this is the translation level, okay, or protein level. Like B cell development. Now let's get into exactly what happens while B cell is developing. First of all, we have the pro B cell, okay? So before this, we have the stem cell. The stem cell can become anything, right? When it becomes a pro B cell, now it's committed to the B cell pathway. So that's why we say this is the earliest cell committed to the B cell lineage. خلاص. Once the stem cell becomes a pro B cell, it's going to continue to become a mature B cell or B cell, right? The marker of pro B cell is CD19. طيب. So CD19 is the marker of B cells because all B cells have to express CD19 since the first cell committed B cell lineage has CD19. طيب. Now this is the level where VDJ recombination and TDT occurs. You can see there's no antibody at this level because it's still being transcribed and translated inside the cell. So this is the stage where this happens. Now, once the antibody is secreted, it then becomes a pre-B cell, okay? This is pre. Yeah, pre is closer to the B cell than the pro, okay? Now, what are the special features of a pre-B cell? First, it expresses the earliest form of antibody, okay? And it expresses it as a BCR, obviously. And what are the components of this antibody? It's a mu heavy chain, and therefore IgM. And then we have surrogate light chains, okay? You don't need to know this, okay? We have light chains that are called surrogate light chains. What does this mean? These light chains are not going to continue with the B cell. These light chains, the function of this light chain is simply to make sure that this heavy chain is properly made, that it can bind another light chain, okay? This is the only function of these surrogate light chains, okay? So it's just to make sure that this heavy chain is going to work properly, okay? There's also two membrane signal proteins, Ig alpha and Ig beta. These are going to be present up until the mature B cells. And this is how, when something binds to the antibody or the BCR, the cell inside knows to activate the process of cytokine production and so on. It activates the cell. How does it know? These membrane signal proteins are connected to the antibody. Once the antibody binds to something, it's going to activate whatever is inside, okay? So this is the structure of a pre-B cell. Two heavy chains that are IgM, mu, and then two surrogate light chains that are going to be the same for all the cells at this stage, okay? It's just to make sure that the heavy chain is working properly. Now, what's the end goal of this whole thing? It's to make sure that the cell can express a functioning a functioning BCR, okay, a functioning receptor. This is, however, a pre-antigen receptor. Yani this is not specific to any antigen. It's just as a test, yani زي test run, okay? So once this binds, if it binds correctly, a signal is going to be sent inside and oh, this one is successful, let's divide it, okay? And this is what we call expansion, okay? So once the signal occurs, this pre-B cell is going to divide into many B cells, and all of these can then go on into the next stage, which is becoming an immature B cell, okay? How does it become an immature B cell? It needs to express the IgM protein, the one that already existed. However, it's gonna make a light chains, okay? And in the beginning, all of the light chains are kappa. Okay, there's no option of the light chain to become lambda in the beginning. However, it can later on switch to lambda if it wants to. However, in the beginning, most light chains are kappa, okay? And it's, as we said, a VJ recombination process. There's no D for light chain. And now the BCR is antigen specific. Okay, so this is pre-antigen, this is antigen specific. Okay, and once the cell has this structure, we call it an immature B cell. طيب. Now, what happens at this stage? Positive and negative selection. What does this mean? For any immune cell, we need to make sure that one, it can bind to non-self antigen, non-self. Secondly, we also need to make sure that it does not bind to self, okay? Because if it binds to self, this will cause an autoimmune disease, okay? So this is a very important step, which is positive and negative selection. So what happens? These stromal cells, or the cells in the bone marrow, present self-antigen. If it reacts, that cell goes to apoptosis. Then it presents a non-self antigen. If it binds properly, it's good to go. طيب. So central tolerance is a word you're going to come across a lot, and it simply means preventing autoimmune disease at the level of the development of cells, okay? So if the receptor binds strongly to self-antigen, it's given a chance to edit the V chain, right? And try again. If it still binds to the self-antigen, or it's not functioning, it does not bind to non-self-antigen, it will undergo apoptosis. And this is the concept of central tolerance, okay? That's it for B cell development. I hope it makes sense. Now let's talk about what happens after the immature B cell. So what happens next? Immature B cells leave the bone marrow through the blood, and it goes to the spleen mostly, okay? And that's where B cells develop into follicular cells. Follicular cells are cells that express both IgM and IgD, and these are naive B cells. Naive means it has not seen an infection or antigen before, okay? So it's naive. And how do you know a B cell is naive? If it expresses IgD. Once the B cell is not naive, خلاص, it won't express IgD anymore, okay? Now, there are two types of circulating mature B cells. B1 is called T cell independent B cells. And these are basically the innate form of B cells. We do not say they're part of the innate immune system. However, their functions are very similar, and they can only produce IgM. And those are the antigen presenting type of B cell, okay? It's B1. B2 are T cell dependent, and they're they're the adaptive form of B cells. They're not the innate form, and they can produce any type of antibody, IgM, IgA, IgG, depending on what the signals it got from T cells. Okay, so that's why we call T cell. That's why we call it T cell dependent B cells. Now, the interaction between B and T cells is gonna come later on, post mid. So you don't need to worry about it for now. Now let's start talking about T cells. Okay. T cells have two subtypes. CD4 plus T cells, that's the marker they have, and those are called helper T cells. Then we have CD8 plus T cells, which are the cytotoxic T cells. Now, all T cells express CD3. So all the T cells will have CD3 on their membrane, and then plus CD4 or CD8, depending on the type of the T cell. Now, let's compare the B cell receptor with the T cell receptor. B cell receptor is basically an antibody that is associated with these signaling proteins we spoke about earlier. T cell receptor is different. It has alpha and beta chains. Okay, it only has two chains, alpha and beta, and then it's associated with CD3, and then either CD4 or CD8. CD3 is instead of these. Right? They're the signaling proteins, and then CD4 and CD8 have different functions that we're gonna touch upon a bit in a bit. Okay. Now, what's MHC? We need to understand what MHC is before we uh, deeply go into T cells. So MHC stands for Major Histocompatibility Complex. Okay. Another name for MHC is HLA. Okay, and I'm sure you went across HLA before. So all the cells in the body have to have MHC, some type of MHC. There's two types of MHC. MHC1, which is it's found in different forms. They're called HLA-A, HLA-B, HLA-C. Okay, and it presents on all nucleated body cells. Okay, every single cell in your body has an MHC1. Okay, except red blood cells because they're not nucleated. It binds to CD8 plus T cells. Okay, in the A3 domain. This A3 domain binds to CD8 in the T cells, the CD8 cells. That's the extra function I told you I'm going to mention in a bit. So CD8, the function of CD8 is to bind MHC. Okay, then the MHC1, the function is to present self-antigen. Okay, it mostly presents self-antigen. That's how the T cells know that this cell is not infected. However, once it presents a non-self antigen or is completely absent, then the T cell knows there's something wrong and then goes ahead and directly kills that cell. Okay, so that's the basic concept. And MHC is found in all cells, and it usually has a self-antigen on it. If a T cell comes and finds that T cells are around sniffing all cells, once it finds its self-antigen, خلاص, it leaves it alone and goes. If it finds it's a non-self antigen or something wrong with this, whatever is expressed on the MHC is gonna kill that cell completely. Okay, so that's how it works. Then we have MHC class 2, and it's symboled as HLA-DQ or HLA-DW or HLA-DR, and there's many types of MHC class 2. They are only present on professional antigen presenting cells. And for the 10th time, the antigen presenting cells are macrophages, dendritic cells, and B cells. Okay, so only these three cells have MHC class 2. And this binds with the CD4 plus cells. CD4 binds with the beta 2 domain. So this is the function of CD4. And it always presents non-self antigens. So after phagocytosis, it presents the non-self antigens that it found. We're going to go up in much more detail in a bit. So this is the basic concept about MHC. Now, we'll talk about exactly how each MHC works, starting with MHC class 1, okay? So what happens is we have a protein inside the cell. This protein can either be virus-specific protein or a misfolded protein or a tumor protein, okay? So once a protein falls under any of these three categories, it's tagged with ubiquitin. Ubiquitin is basically a tag that tags cell to death. It tags proteins to death, okay? So once this protein is tagged, it goes to a protein complex called a proteasome. Proteasomes are the site of ubiquitin tagged cells breakdown. So once a protein is tagged with ubiquitin, it goes to the proteasome, and this cell gets broken down into these, let's say, antigens. Okay. Now these antigens then go to the endoplasmic reticulum. Okay. In the endoplasmic reticulum, there is constant production of MHC1. It's always being produced in the endoplasmic reticulum. The antigen is transported into the endoplasmic reticulum using TAP protein, and then the antigen is loaded onto the MHC using tapasin protein. These two proteins are very important to remember their names. Then once the antigen is loaded on the MHC, it's gonna get transported to the Golgi apparatus via vesicle, And then from the Golgi apparatus through exocytosis, it's going to be exocytosed to the cell surface membrane. And then this cell is going to be presenting this antigen of either the virus or tumor or whatever. And then the CD8 plus is going to recognize it, going to bind to the MHC, and then kill the cell. Okay, that's how it works. This is what I said in words. Now, viruses can sometimes escape being detected in some ways. Firstly, it can remove the newly made MHC from the ER. Okay, it stops the process over here, or it can prevent the maturation of this MHC, and if it did not mature, it cannot take the antigen, so it can stop the process here, or it can inhibit the transcription genes of MHC to begin with from the nucleus, or it can block this peptide, so the antigen can not go into the endoplasmic reticulum. So the viruses are smart and they can evade this process in many ways. How MHC2 gets loaded. Now, remember, MHC2 is only an antigen-presenting cell, so phagocytosis has to occur in order for MHC2 to be able to present antigens. Now, the goal of this process is to identify bacterial antigen by CD4 plus cells. So what happens is, once an endosome comes in after phagocytosis, it binds or it fuses with the lysosome, and this is now called a phagolysosome. And in the phagolysosome, the bacteria is broken down into small proteins or antigens. Okay. Now, in the endoplasmic reticulum, again, MHC2 is being constantly produced. But however, when they're produced in the endoplasmic reticulum, they're always bound to a protein called the invariant chain. The invariant chain, at the end of it, there's this part called clip. Clip is very important in order to not allow stuff that's supposed to go to MHC1 to bind to MHC2. Okay, so it's basically a lock for now. I don't want anything to bind to MHC2 while I'm in the endoplasmic reticulum. Once the phagolysosome happens, MHC2 travels through the Golgi apparatus to the phagolysosome, and it incorporates into the membrane of the phagolysosome. It then associates with a protein called HLA-DM. HLA-DM has a very similar structure to MHC2, and the function is to replace this clip protein with the antigen. Once the clip protein is replaced with the antigen, the MHC2 is released from the phagolysosome and to the cell surface, where it interacts with CD4. Then CD4 produces cytokines in order to activate B cells to produce antibodies against this specific bacteria. Okay, and this is what I said in words. Okay, now we're gonna take a short salah break and then come back and continue T cell development. We're almost done. There's this lecture and then a very short lecture 8, and then I'm gonna do lecture 5, which is complements. Okay, so we stopped at T cell development. So as we know, the T cell development happens in thymus. The thymus structure, there's a cortex, there's a medulla, and then other than thymocytes, which is another name for immature T cells, there's also stromal cells, and there's also dendritic cells, and there's also macrophages. So what are the functions of each of these? First of all, in the cortex lies the immature thymocytes. The cortex is the region nearest to the capsule. Okay. Then in the medulla, we have the mature thymocytes. So as the lymphocytes mature, or the T cells mature, they travel towards the center, and that's because the venules that will take the mature T cells into the lymphatic system are in the medulla. We're going to talk about this in a bit. Then stromal cells, their functions is first of all positive and negative selection. So they present self-antigens to thymocytes, the same way we mentioned how B cells develop and they need positive and negative selection. The same thing happens to T cells. in the development. And the stromal cells also produce IL-7, which is an important growth factor for T cells. Then we have interdigitating dendritic cells that also present self-antigens to thymocytes. Then we have macrophages, which phagocytose apoptotic thymocytes. So the cells that failed the test are going to undergo apoptosis, and they're going to be eaten up by macrophages in the thymus. So what are the stages of maturation? There's much more detail into this. However, it's not included in your midterm. So simply, there's TCR or T cell receptor recombination. And this happens via VDJ recombination for beta chain and VJ recombinations for the alpha chain. So as we said, the structure of T cell receptors is different from the B cell receptors. You can think of the beta chain as the heavy chain and the alpha chain as the light chain. So for beta chain, there's VDJ because it's heavier. And for alpha chain, there is only VJ. And the process is exactly the same as what happens in the B cells, this VDJ recombination. Now, then happens positive and negative selection or central tolerance, as we mentioned. And then, just to be clear, central tolerance only refers to the negative selection, not to the positive selection, okay? Just so you don't get confused. Now, the mature lymphocytes, these ones, then travel through the lymphatics, these venules, into the lymph nodes. And as we mentioned, the lymph nodes are the secondary lymphoid structures that are responsible for activating these mature, naive T cells. Naive, again, means that they have not seen any infections before. And how do the thymocytes or the mature T cells know their way to the lymph nodes? They express something called CCR7, which is a chemokine receptor. And as I'm sure you know by now, chemokines are responsible for migration of white blood cells into the area that they need to go to. So CCR7, the chemokine receptor, directs their migration. And this migration is called diapedesis, okay? Now in lymph nodes, they can get activated through antigen presentation. And once they get activated, they're now called effector T cells. They're specific to one antigen, and now they can go to the blood. They divide first of all, and then they go to the blood to encounter the pathogens, whether they were cytotoxic T cells or helper T cells, okay? And now the extravasation, so from the blood to the tissue, happens through integrins and selectins, and those are called addressins on the endothelium, and certain molecules on the leukocyte as well. Now, you don't need to know this in detail. However, you need to know it in detail for acute inflammation in the pathology lectures, but not for immuno. Now, to talk simply about chemokine, again, you need to know about chemokines in detail in the pathology lectures, not here, or not yet, at least, the immunology course. You have two main chemokines, the CC and the CXC family. C is for cysteine, okay? So this means that a cysteine is connected to another cysteine directly. CXC means that cysteine is connected to amino acid, random amino acid, then connected to a cysteine residue. And this is the chemokine of neutrophils, mainly. Another name for it is IL-8. This is important. IL-8 or CXC is the main chemokine for neutrophils. CC is the main chemokine for macrophages. Both CC and CXC can call lymphocytes, okay? And they bind to CCR7, which is the chemokine receptor, okay? Chemokine secretion is stimulated by inflammatory cytokines, namely IL-1, IL-17, and TNF-alpha. This is a very repetitive concept that IL-1, IL-17, and TNF-alpha are the acute inflammatory cytokines. They're not important in immuno, however, they're extremely important in pathology. They're also going to be important later on for post-midterm lectures for immuno. That's it for lecture 7. Now, this is a very easy, however, it's an extremely important lecture. Very high yield, extremely high yield. Alhamdulillah, it's very easy. How do these naive T cells, once they go to the lymph nodes to get activated, how do they turn into effector T cells? Okay, what happens is you need 4 requirements. All of these 4 things need to be present in order for a naive T cell to turn into an effector T cell. Okay, the first thing is obviously the T cell receptor binding to an MHC with an antigen, okay? So if the T cell receptor is complementary to this antigen, so if you remember, each receptor is specific to one antigen. If it finds that antigen, it's going to bind to this MHC complex through either through CD4, right? And the second condition is CD28, CD80, CD86 binding, okay? So CD80, CD86, other name for these proteins is B7, okay? So these are B7 proteins. CD80 is B71, CD86 is B72, okay? These are found in the antigen-presenting cells, and this needs to happen. This binding needs to happen in order for the naive T cell to get activated. Another thing that needs to be present is the ICAM LFA1 interaction. This is an integrin. Whenever integrins are involved, this means the function is adherence. So in order for the thymocyte to adhere and stick properly to the antigen presenting cell, in order for this all to happen, you need an integrin LFA1 interaction. And the fourth condition is cytokines being released from the antigen presenting cells into the naive T cells in order for them to proliferate, okay? They get activated. And the main interleukin for T cell proliferation is IL-2. This is also an important piece of information. So this is what I said in words. It's important to know, move us to know these four conditions. It's very important to know where each thing lies, okay? So CD80, 86, CD28, okay, you know it's a requirement. But you also have to know that CD28 is part of the T cell and CD80, 86 is part of antigen presenting cells, and so on for the rest, okay? So it's very important to know with what is where. But other interactions that can happen between antigen presenting cells and T cells. Number one is downregulation. So let's say an immune response happened and you want it to stop. You don't want the immune response to go on for very long because it's damaging for the body. So what happens is two things. The first thing is that T cells start to express CTLA4, okay, which is basically the replacement of CD28. However, this does not activate the cell. Right? So this is one way, competitive inhibition of CD8086 by CTLA4. That's one way. The second way is PD1 ligand. Okay, and what happens here is once inflammation starts, effector T cells start expressing PD1, and then on the antigen presenting cells, there's PD1 ligand. And PD1 ligand binds to the PD1, it inhibits the action of the T cell receptor. So even if the T cell receptor binds, the intracellular signal is not going to work, okay? Because there's CD3 here, and then there's a tyrosine kinase that will activate intracellular pathways. All of this you're going to know later on. However, this whole process does not work when PD1 binds to PDL1, okay? So these are two ways to downregulate and stop the immunity from continuing, or the inflammation from continuing, okay? It's also important to know what is where. Again, the PD ligand is on the APC, and the PD receptor is on the effector T cell. And then finally, what if I want to increase the immunity? Okay, this is for downregulation. What if I want to increase it? The APC and the effector cells can actually talk to each other. And this is through another reaction called CD40 ligand. This time the ligand is on the T cell and the receptor is on the APC, unlike PD1, okay? So what happens is, once this interaction happens, three things happen. An upregulation of CD80, CD86 on APC, okay? So this becomes more, and therefore more activation happens. This leads to the upregulation of MHC expression, and this leads to the upregulation of cytokine production. So overall, it increases the immune response. So these two slides sum up the whole lecture, and they're very, very important, right? So lastly, the complement system. Extremely easy lecture. It's pure memorization, so you need to memorize well. Like, complements are a group of proteins and enzymes made in the liver. They circulate as zymogens, which, as I said, mean pro-proteins or pro-enzymes. So they need to be cleaved in order to be activated. And that's why you'll see a lot of cleavage in this lecture. It's not found in the CSF and urine. Honestly, this should have been very small font, not important. But what are the functions of the complement system? Number one, opsonization. If you recall, opsonization means tagging cells in order to make it easier to phagocytose. Okay. And the main complements that are responsible for opsonization are C3b and C4b. This is just memorization, that's why I'm going fast. They can also act as anaphylotoxins, which means they can induce allergic reactions. And they do that by binding to mast cells, causing histamine release and vasodilation. And this is what we call anaphylaxis. They can also work as a chemotaxin, and the main chemotaxin is C5a, and it also stimulates the respiratory burst in oxygen-dependent killing. Okay, and then they're also responsible for MAC formation, which is membrane attack complex. We're going to talk more about this soon. Okay, now in order to form a membrane attack complex, there are three pathways, and all of these three pathways lead to the formation of a membrane attack complex, which basically kills the cell directly, right? Kills cells. Like, how do these complement system get cleared from the blood? There are receptors on the RBCs that take C3b and C4b. C3b is the most important complement, and we'll get to know why in a bit. And it goes to the liver and spleen, where their whole red blood cell with these complements is phagocytosed and broken down. Like, these three pathways are the most important thing in the lecture. So what are these three pathways? So the first two pathways, the classical and the lectin pathway, they start by actually the classical pathway starts by an antigen antibody complex. If you recall, in antibody killing, we said the third way is through the complement system. And this is how it's done, okay? Once an antibody binds to an antigen on a cell surface membrane and forms a complex, complement 1Q, okay, binds to this complex. And once it binds to this complex, it opens in the same complement 1, it opens C1R and C1S domains, which are serine proteases, okay? Proteases are cleavers, they cleave stuff, right? Once these domains are exposed, they cleave C4 and C2 into C4A plus C4B, and then C2A plus C2B. However, we're only going to focus on what's important from this product. The important is C4B and C2A. These two combine in order to form C3 convertase. C3 convertase then cleaves C3A into cleaves C3 into C3A plus C3B. However, the important one is C3B. Okay. Now C3B goes back and joins this complex, forming C4B, C2A, C3B, and this is called C5 convertase. C5 convertase cleaves C5 and forms C5B. C5b is the initiating complement for membrane attack complex. It goes and binds to the membrane of the cell or the bacteria, and then it stacks up the rest of the complements, C6, C7, C8, C9, and all of those together form the membrane attacks complex, which basically forms a pore or a hole inside the cell membrane. Once a hole is done, the cell is going to burst and die. Okay, so this is the process. I'm going to repeat it in writing. So C1q binds to the antigen antibody complex, exposing C1R and C1S. These are serine proteases. These cleave C4 and C2, leading to the formation of C4b, C2a complex. This complex is known as C3 convertase. C3 convertase cleaves C3 into C3b. C3b then goes and joins the same complex to form C5 convertase. C5 convertase cleaves C5cb, cleaves C5 into C5b, which initiates the formation of the membrane attacks complex. Then C6, 7, 8, and 9 join C5 on the membrane, leading to the pore formation and cell death. Now, what can they ask? They can ask, well, what is C5 convertase? Which of the following is C5 convertase? You need to know that this is C5 convertase. They can ask plenty of stuff. I remember we had a very silly question saying, how many proteins are present in C5 convertase? So it's 1, 2, 3. Okay. So this is how they can ask. What about the lectin pathway? Lectin pathway is exactly the same. However, only this step is different. So from 1 and 2, from 3 onwards is the same. 1 and 2 is different. What's 1 and 2? Instead of C1 being activated or C1R and C1S cleaving, we have Manan binding lectin, which we spoke about in the beginning of the lecture. It's an acute phase reactant, and it forms a complex called MBL MASP complex, and this is what cleaves C4 and C2. Okay, so this is the only difference. Instead of this, we have MBL MASP complex. Okay. Now, finally, the alternative pathway. This is completely different from the other pathways. So this is initiated by an infection. Once you have C3b from any source, okay, already activated C3b, when it finds a microbe, okay, it binds with this microbe, forming a C3b complex, okay? Now, we have something called factor B, which is cleaved by factor D into BA and BB. Our BA is not important. We're only focusing on BB. BB joins C3b complex, forming C3b-BB, which is also known as C3 convertase, another type of C3 convertase. And then, in order to stabilize this is not a stable. This is not a stable enzyme. In order to stabilize it, a factor P comes and joins. So now we have C3b, BB, P. This is a stable form of C3 convertase. So this then, another activated C3b joins and forms C5 convertase. And then the same happens. Well, C5 starts the MAC, and then 7, 8, 9 join to form the MAC complex. Okay. This is pure memorization. It's a lot of weird names. This can get confusing. Wallah, look at this here. Another question that they can ask, C5 convertase from the classic pathway is only 3 proteins. Like C4b, 2a, 3b. Only 3 proteins. However, here it's 1, 2, 3, 4. It's 4 proteins. So this is another question they can ask. And that's it for the lecture. Thank you for listening. If you have any questions, please don't hesitate to ask. I hope I didn't forget anything. Please go through the slides quickly just to make sure that I didn't miss anything and that you understood everything. And if the questions are going to come from your slides. I hope it helps, and thank you. Good luck on your midterm.